It's our pleasure today to introduce the co-founders of JoVE, the Journal of Visual Experimentation. I'm Kat Allman with the Open Source Programs Office at Google. We are very pleased to have these two gentlemen with us following their participation in O'Reilly ScienceFoo Camp held last summer here at Google. They've come today to talk about a new method of publishing scientific, specifically biological, research and methodology. Without further ado, Moshe Pritsker and Nikita Bernstein.
Hear me well? All right, it's a pleasure to introduce JoVE for this massive audience today, and especially that half of the audience in this or another way we met before. Again, my name is Moshe Pritsker. In JoVE, I'm responsible for the science part, and Nikita Bernstein is our computer person, so questions about computers to him, about science to me. So JoVE is a video publication for biological research. And today I'm going to tell you briefly what you will hear today is about the idea why to publish research on video. Then about the execution, how to do it. Our business model, people behind it, and why do we think it can be interesting to Google since we are here at Google. So I will start immediately from the statement of the problem. There is a huge, huge problem in biological research, traditional problem existing for hundreds of years. It's very difficult to reproduce experiments in biology. So usually, and without reproduction, imagine yourself two different teams at Google when one of the teams cannot reproduce what is done by another team. In biology, in biomedical research, it's typical, both in academia and in an industry. In time, it takes between few weeks to few months to reproduce an average biological experiment, which can be published and described maybe 10, 20 years ago. And here's here's a good example of how it looks in reality. So this is a typical experiment in stem cell research. Basically, this experiment is is at the foundation of the cloning approach, and it's an experiment about enucleation of mouse oocyte, where you have mouse oocyte single cell, and you're supposed to take a nuclei out of this cell. And look at the description of this experiment as typically given in scientific journal by the group who managed to master this experiment in their lab and now try to convey how they did it to scientists. Position the metaphase spindle at 3 o'clock and hold it with a holding pipette. Apply piezo pulses to penetrate the zona pellucida. Tie the metaphase plate with this nucleation pipette. So you can read it few times more. And it doesn't matter how many PhDs in biology you have. In general, it's very, very difficult to reproduce this experiment. And even to understand this experiment, just looking at this description. And so how it is done in reality? In reality, this experiment looks like this. So this is your oocyte. This is your two pipettes. One of the pipettes goes inside of the cell, takes away the nucleus, and basically spits it out. Very simple, right? And that's a problem in today's biology. The concepts in biology are so simple. You can take everyone from the street, explain them what is the cell, what's the goal of the experiment, and they will understand it. Yet to understand the description and repeat it in your own lab, and doesn't matter how many years of training you have, is very difficult. And here we look at the consequences of this problem. So the printed scientific literature, for some reasons, I will discuss them in a minute, for some reason does not provide an adequate description of biological experiments. Then we have a problem, an inadequate knowledge transfer in science. Well, from here, we have low efficiency of biomedical research. If we have to repeat the experiments again and again and again, then what kind of efficiency we are talking about? And from here, in the end, it leads you to this high cost for development of new therapies. It takes about $1 billion and 10 years to develop a single drug. Not all of this is coming from this problem. but significant part of it is coming from this problem. So being myself in the lab for many years and suffering personally from this problem, I began to think about the solution. Just to illustrate the program once again, one day in my lab, I tried to repeat experiment on stem cells, which was published 2 years before in a very high-profile scientific journal by the group whom I knew personally. And I was not able to do it. No one else in the lab was able to do it. My boss at Princeton, where I did my Ph.D., sent me to Scotland, where that lab was working and doing those experiments. And that's typical in biology today. You really want to learn the experiment, you either spend a few years of your life, or you go to that lab if you have enough money and they're kind enough to accept you, and you learn how this experiment is done. And then I began to think, so what would be the ultimate solution of this problem? So the ultimate solution should mimic the process which happens today in the lab. You want to learn experiment, someone has to show you. So show in our age means video. So the best solution would be to have a huge database where you would have 1 or 2 videos for every possible biological experiment. And so if we will have such a database of videos, then we will increase the reproducibility of the experiments and increase the efficiency of biomedical research across all the areas. So, OK, to build such a database, let's think how to build such a database. We have to show lots of biologists. We have to have lots of biologists showing their experiments. And they have to have an incentive to do that because they are busy people. They are working on their careers. What would be the incentive? And then I realized we should not call our database. Our database should be called journal. Because journal, scientists work for publications. All their life is about publication. They get recognition based on publication, professional promotions, and grants based on their publication. So what if instead of database, we will found a journal? And so we came with this idea of journal of visualized experiments. I think it's better to show you now on how it looks in reality. Uh-oh. OK, so this is our journal of visualized experiments, where basically videos, or we call them video articles, because again, we are now a publication journal, we are not database. Video articles published on different types of experiments. For example, these video articles will tell you how to do immunohistochemistry, or in simple words, identification of different proteins in particular mouse tissues. So let's look how it looks. Just to mention that all our material is open access and publicly available online. And I will touch on why is it and our business models a little later. So, and basically, our videos very much modeled after traditional scientific articles, having an introduction and having a...
In the epidermis, immunohistochemistry is an efficient means of localizing specific proteins to their expression compartment. In this video, we demonstrate a reliable method for immunolocalization of protein in the epidermis, which we use routinely in our laboratory.
So that was now we are going into short introduction where scientists would introduce...
Hi, I'm Tammy from Dr. Turkson's laboratory here in the Loeb building of the Ottawa Health Research Institute in the Chronic Disease Program. Today we're going to show you...
So experiments shown by scientists themselves, by scientists who know to do these experiments, because, I mean, they're the best source of knowledge. And again, we model these videos more or less following the current format of the traditional scientific publications, where you have introduction, you have description of the experiment, and you have a sort of discussion. What's happening? OK. And so, starting from introduction,
use a core facility.
We go into the real experiment.
After receiving the slides back from the core facility, you can see that the paraffin is present on the slides as a thin film. The slides are placed on a rack, wrapped with aluminum foil just like a baked potato, and incubated for 5 minutes at 55 degrees.
So, and here you can see how video is able to reduce all these complex scientific descriptions to very simple, basically cooking methods. As she describes it, take this slide, do something, put it as a potato into the microwave. But it is indeed how science is done in the real life. So in a sense, we give, in addition to increased reproducibility of the field, it also makes science more accessible to wide public. Now, coming back. So a few words more about the journal format. So as a journal, you are supposed to have editorial board. So our editorial board is about 20 professors from Harvard, Princeton, NIH, and other places who basically were excited about the idea, excited enough to put their name on our journal for free. Their main function is advising and editorial advice to the journal. So, okay, how it all works. Starting from science. Again, academic journal. As academic journal, you're supposed to have editorial board. We have it. As academic journal, you're supposed to have sort of review. We have it. And you have to be in a PubMed. So PubMed is a little bit more difficult process. PubMed is the database, basically list of journals maintained by National Institute of Health. And in order to be recognized as academic journal, you're supposed to be listed there. But here's the problem. They never had a video journal because all the journals today in biology are in print. But after some negotiations and talks and suggestions, now we are undergoing the review, and in June we're expecting the final decision. So hopefully we will be there. How this video is made? Initially, we thought it will work like YouTube. People, scientists will make their cute small videos and will send it to us and show their experiments. In practice, it did not happen for the only one reason, in my perspective. It's very difficult to make a video on biological experiment. It's much more difficult to make a video on biological experiments than make video of myself singing guitar, simply because requirements for editing and filming are much higher for complex processes such as biological experiments. So scientists don't have software to edit the experiments. They don't have time to learn this software. They don't have good cameras. So what we have to do is to provide technical help with filming and editing to their labs. But here's the problem. Their labs are spread across the world. The two biggest hubs of biotech, for example, in the US, in California and Massachusetts. So in this situation, what we have done to deal with this problem, we established this distributed network of video production where we have filmmakers who also have certain experience. We screen them so that they would have a certain experience in biological scientists, and they would have experience in biological sciences, and they have a certain, they work with, on the contract with us. So every time when we need to film something, we pay them, they go, they film, they give the material to us in edited form. It undergoes approval, reviewer's approval in our journal, it's approved by scientists, and then it is published as video article in the final form that you saw. Few other aspects, so web technology. Nikita took care of the web technology using the platform developed at the company he worked before. It's called CMAX, and it basically graphic-based database platforms that allows him to maintain all our complex site by himself. On video online, we outsource our video from Videotac. For the business model, we initially wanted to make our video articles open source. We wanted it to be available to all the scientists. So we could not use a traditional subscription-based model of scientific journals. Instead of that, we are using advertisement-based model where we expect that people who watch our videos are scientists, and therefore our journal provides a platform for highly targeted advertisement, first of all, for producers of biological tools and equipment. and it's quite a big market. So financing. Financing came from angels. So these are points at different complex components of JoVE I mentioned to you. Science, video production. As I mentioned to you, we have now a video production network established in 20 cities across the entire United States, but also in Tokyo, Berlin, Canada, and other places. Web technology, Nikita can tell you more about this. This is how the platform looks and how he implements it, the design of our site. The business model. Again, JoVE receives content from scientists, and JoVE would receive advertisement from producer of research tools. And scientists, by watching JoVE, they also get experience with these research tools as they shown in video on JoVE. And this circle would establish a basis for profitability of our venture. Here's the statistical data for the market. OK, how now, today results, where are we today? After 1 year of work, we published 200 video protocols, mainly produced by labs at best US and world universities from Harvard, MIT, and etc. So this, we received quite a lot of recognition in the scientific community. For example, our articles now cited by other scientific journals. And our idea, again, is to continue. And we basically remain a single video journal because our approach is quite novel, and it's difficult for existing scientific companies to integrate all these new components like video online, distributed video production, web technology. So meantime, we happily enjoy this position on the market. The team, myself, Nikita Bernstein, Klaus was our financier, and Aaron Kolskandriako took care of our video production. Handen and Dita, Christina, and Anna work mostly on the editorial part. All of them are biological scientists. So, and the last part of my talk, why I think we can be useful to Google. So at Google, I know you have a couple of initiatives that everyone is aware about. One of them is Google Scholar. So what you have in Google Scholar is a massive repository of scientific articles. Now, our videos show methods that are used in the scientific articles. So potentially, each of these scientific articles, if linked to these videos, can benefit, because this video shows how you do the experiments that are described in the scientific articles. Therefore, it can make Google Scholar more useful and give certain advantage to Google Scholar for scientific community. Well, obviously, connection with YouTube and Google Video. And Nikita, would you like to tell a few words about Open Social and AdSense?
Sure. So basically, our understanding is that there are a lot of initiatives in Google with respect to social networking. And one of the things that, I'm so sorry. Sorry. So my understanding is that there are a lot of initiatives at Google with respect to social networking. Open Social made a big splash. And we see ourselves as a potential channel to reach the academic and biological industry audience. So we're kind of curious to find out whether we could mutually benefit from using Open Social in our context. With respect to advertising, just a couple of words to add. The reason that we feel that advertising model makes a lot of sense in our case is because of really, really high cost of equipment and the difficulty for industry to reach their target market. So we are interested in looking at how we can leverage AdSense to help us connect with the market and possibly keep our internal costs down with respect to the advertising platform. Yeah.
Just to summarize, again, we combine few very different components: science, web technology, video online, scientific publishing model, in order to create this new method of scientific publishing, video publication. I think we are quite unique in this sense, and we are happy and open to hear any ideas and explore any possibilities for collaboration, partnership, etc. Thank you.